this local selection may be kind of counteracted by gene flow from elsewhere. So what is your idea? What kind of gene flow might be taking place between these two bays in copepod populations?
I don't really understand the question.
New
Can you
genetic.
speak more into the microphone? I can't hear you.
There may be new genetic alleles coming from elsewhere to these bays, and if that is the case, if there is lots of gene flow, there can't be much of local adaptation taking place.
So you mean other copepods swimming into the bays?
Exactly, yeah.
I think it is unlikely in the heated bay because of the water flow, because there is a constant current like out of the heated bay, so I don't think copepods can go against the currents.
Yeah. How about, can they survive through the nuclear power plant system?
Yeah, it has been measured that there is very, very, very few copepods coming from the water itself. So actually, it's not a problem because there's so few.
In the Baltic Sea, we don't have tide, but we have quite a lot of water level fluctuations that are related to wind-driven currents and related to water pressure differences among different parts of the Baltic Sea. So these water level fluctuations will transport huge water masses along long distances in the Baltic Sea. So I would guess that copepods that are kind of sticked into the water column, they move with these water currents, and therefore they can move from one bay to another, or from the pelagic areas to the bays, or vice versa.
Yeah, of course, if they are in the water that is inflowing, I haven't really thought about that. But of course, then if they are gonna mix with the copepods, then there will be lesser chance. If they are mixing with the copepod that is already in the base and have been infected for a long time.
Exactly. That would, if that is the case, it would make this microevolutionary change less likely.
Yeah.
Okay. Then a question related to, somewhat related to this thing. In the first paper, in the figure 2, that you already showed in your initial lecture, you nicely summarized the differences in abundance of copepods between heated and control bays. But I had an idea, or I understood that you interpret it as like species having different reproductive cycles in these two bays.
So the reproductive cycle, the cycle is that they make the egg either dormant or immediately hatching. It hatches, it becomes a napoli, it becomes a copepod, they make an egg. It's the same. Yeah,
I'm kind of thinking that there were some differences in the species copepod species composition among the bays, wasn't there?
Yeah, it's in the supplementary.
Yeah. So in the warmer bay you had Acartia and Harpacticoids, and in control bays Cyclops and Eurythemora species. I think that those were the main differences.
Yeah.
So could it be that these differences that you here show, for example, considering total abundance, that they were mainly due to the differences in species composition among the bays, but if you would look at certain specific species, would they still be different, these abundances, or would they be more or less the same?
We have a figure in the supplementary where we look at different species, and there are different curves for the different species. I can find it. It's do you see it?
Exactly. Okay. Yeah.
Yeah. So we looked at different species. So we can see, for example, cyclopods. They were lesser in the heated bay than in the control bay, meaning that they are more adapted to a colder environment. Is that your question?
Yes. So okay. Yeah, now I got. So you had species that responded differently in bays, and then you had also species that showed more or less the same kind of dynamics in both bays.
Yes.
Yeah. Yeah. Okay. Good. Maybe then one question about was it the second paper? Yeah, and congratulations that you got it published already.
Thank you.
I wonder whether the reviewers said anything about one detail in the very first paragraph about the claim that coastal areas provide nearly half of the global oceanic primary production.
Yes.
Did the reviewers agree with that?
I don't remember there were any problem with that, but what's the problem here?
Yeah. I was wondering that a little bit. If you think of the global primary production in whole Earth, how much is the marine primary production?
About
or
half.
About a half.
Yeah.
Then thinking of the marine primary production.
Please hold the microphone so I can hear you better. I have problem hearing you.
If you think of the marine global primary production, how big a part of that is produced in the open ocean?
It must be about half, because about half is coastal area.
Okay, yeah. I looked a little bit into that, and I found numbers that in open ocean it would be from 60 to 70% of the marine primary production. Then in upwelling zones it should be something like 10 to 20%. So then that calculation would leave like 10 to 30% of that in coastal areas.
Yeah.
Yeah.
Okay.
Yeah. Yeah. Then the figure 2 in this same paper 2, you have in the figure 2 you have the Shannon diversities for prokaryotes and microeukaryotes. Microeukaryotes. So I was thinking that the Shannon-Wiener diversity of prokaryotes is several orders of magnitudes higher than for microeukaryotes, and that led me to think of how many species you have, or what is the species richness or taxon richness of these two groups. It kind of appears that it has to be much higher for prokaryotes than microeukaryotes.
So how many species was
How many species? Or you had these amplicon sequencing variants.
Yeah.
So how many of those you had in as prokaryotes and in microeukaryotes?
I don't remember the numbers.
Yeah, but it must have been a high number, really high, because if you think of the Shannon diversity index that is around 6, I guess it indicates that it can be up to hundreds of thousands of taxa there. So it must have been a lot
higher.
Yeah. Okay. Maybe it would have been interesting to plot that as well.
How many? Each?
Yeah, like the species richness in these different taxonomic groups and in different bays. Yeah. So you have something about this in the figure 4, where you have those Euler diagrams that illustrate the numbers of these ASVs. I found it a bit difficult to understand what are the percentages there. So in the size of the circles is related to total number of unique taxa, yeah, and that is clear. But then, what are the percentages in there?
So the percentage is what is unique. So if you look at the first one, Soma prokaryote, 2% of the taxas is unique to the heated bay, and 5% of the taxas is shared between heated and gradient, and 2% is shared between heated and control bay, and 7% is shared between gradient and control bay. So it's what is unique and what is shared.
But these percentages don't come from these numbers, these absolute numbers of ASVs.
What?
They don't come, the percentages, from the absolute numbers of ASVs.
No, it's unique.
But the title text says that this is illustrating the number of unique and shared ASVs. So, for example, in the first figure, in the heated bay, the 2,701 is the number of unique ASVs.
Yeah, and that is 2%.
It's 2% of not of the unique ASVs, but it's 2% of all the ASVs, including the non-unique ones, isn't it so?
No, what is here is like 2% is unique only to the heated base and not shared with the other environments.
I think I don't get it because
Okay.
to me the percentages and the absolute numbers tell a different story.
Oh, okay.
But the story what it is telling, how I got it was that there are in the middle, in the gray area,
shared.
there are very few unique ASVs, but still they are almost 80% of all the ASVs.
Yeah, so this is what is shared between all the environments.
Yeah, yeah. So is it then so that those are those 76% in the case of the first figure, they are the common species that are very generalist ones. They are found in all environments and on almost all stones you collected. And then there is a very small proportion of the rare species that are kind of could be phrased as being specialists. Specialists for the warm bay, for the control bay, and for the gradient bay.
Yes.
Yeah. So I think that is a very common phenomenon in when one is studying community structures. That in almost all kind of communities there are lots of rare species and very few but widely common species, and this seems to be the case also in these communities. To me, this was absolutely the first data I have ever realized of microbial communities and their structure. So it was very nice result. Maybe it could have been discussed a bit in the discussion, but well, anyway, it's published already, so that's for later discussions.
Okay.
Maybe one more question, and this is about paper number 4.
What? Paper number 4?
Number 4, yeah, the sediment paper, and I was wondering a little bit of this one supplementary figure, supplementary file S7.
S7.
Where where you have done really great work in deriving these rarefaction curves for each of your samples.
Mm-hmm.
And that's really a nice and convincing way to derive the species richness or taxon richness.
Mm-hmm.
So I'm happy you did it. But the thing I'm wondering here is that when you are looking at the time zero, the first sampling, in the uppermost figure,
mm-hmm.
The numbers of taxon or taxon richness varies from about 500 to 1500
Mm-hmm.
ASVs, while during T1 the variation is from about 1000 to 4500. So widely different numbers of taxa sampled during the same season but different years. So where might that difference come from, and do you think that it may somehow affect on the result that you are showing in the figure 8 of that same paper, where you very nicely show the differences in different bacterial groups between control and heated base, and between the resident ones and translocated ones. So my question is that is this difference in the numbers of ASVs determined in two different time points, is it some sort of artifact or difference in the method used?
No, it's just different between the years. Difference, is data how it is.
Okay, so yearly differences in the amounts of taxa are huge.
Yes, I know.
Yeah. Well, interesting result. Yeah. Good that it wasn't an artifact of some method thing. Okay, thank you very much.
Thank you very much. Then we have a final opportunity for the audience to ask any questions, and Linnea can also check online whether there is any questions. It's a forest of hands. No? One.
Okay.
Thank you. So regarding the copepods,
Yes.
In your base,
Yes.
I was wondering, did they freeze in the winter time, the base, and
Yeah.
that influenced the productivity of eggs?
So the heated bay never froze. It was always nice and warm. But the control bay in the winter, there was an ice layer on the water. So there was differences in that way.
Did it influence the egg production, do you think, during the winter?
Not much.
Thank you.
Okay, then I think I will now declare this session closed. So the committee and the opponent and the supervisors will follow me and Linnea to a separate room. And for the rest of you, I think there may be snacks, if I'm not misunderstood. So thank you very much, everybody, for coming here, and I think we end with an applause.